Hello everyone. Today's video's topic is secondary hemostasis or blood coagulation. This is the part 1 of the video which covers still the different kind of blood coagulation factor. In the second part, I will explain the mechanism of blood coagulation. Secondary hemostasis definition. Secondary hemostasis is defined as the formation of insoluble cross-linked fibrin by activated coagulation factor, especially thrombin. Fibrin stabilizes the primary platelet plug, particularly in larger blood vessels where the platelet plug is insufficient alone to stop the blood hemorrhage. The goal of secondary hemostasis is to stabilize the platelet aggregate by weaving a mesh of fibrin to cement the soft clot. The classic coagulation cascade helped to maintain this role. What is blood coagulation? It is the process where the blood loses its fluidity and becomes jelly-like mass. It happens mainly due to the formation of fibrin. Blood coagulation occurs in three stages. First is formation of prothrombin activator, that is factor 10 or X. Conversion of prothrombin to thrombin, second step, and third step is conversion of fibrinogen into fibrin, which forms the heart clot. Generally, the blood coagulation happens with the help of blood coagulation factors. They help they circulate in plasma in a very low concentration. The amplification capacity of cascade reaction mechanisms ensure an efficient response. More than 20 blood coagulation factors are available. Among them, 7 of the coagulation factors are zymogen. Zymogens are inactive enzymes which needed to be activated. These examples are prekallikrein, prothrombin, which is factor 2, proconvertin, which is factor 7, Christmas factor, which is factor 9, Stewart-Power factor, that is factor 10, plasma thromboplastin, that is factor 11, and Hagman's factor, that is factor 12. These all enzymes are belongs to the family of protease enzymes. All these enzymes have an essential serine group in their active site. So here is the list of clotting factors given. The first clotting factor is fibrinogen. It is a protein of 340 kilodalton. It is converted to fibrin by thrombin and helps to form a net that traps the blood cells. Second factor is prothrombin. It is a 72 kilodalton protein. It activated to thrombin by factor X. A. The third factor is thromboplastin. It is a 44 kilodalton protein. It is released when there is a cellular damage. It participates in factor 10 activation. The fourth factor is calcium. It mediates the union of factors 9, 10, 7, and 2 with phospholipids, which is present on the surface of platelets. Fifth factor is proaxillerin. It is a 350 kilodalton protein. It potentiates activated factor 10 action on prothrombin. Factor 6, it actually doesn't exist. Factor 7 is proconvertin. It is a 45 to 54 kilodalton protein. It forms the complex with factor 3 and calcium that activate factor 10 in extrinsic pathway. Then factor 8. It has two components. One is antihemophilic factor, that is factor 8C, Another one is Van Willebrand factor, that is factor 8R. Factor 8C, that is anti-hemophilic factor, has a molecular weight of 285. It associates to factor 9A and helps in the activation of factor 10. Its absence causes hemophilia A. The second is factor 8R, that is Van Willebrand factor. It is a big polymer of around 10,000 kilodalton. It mediates the union of platelets to collagen in vessels subendothelium. Its absence causes Van Willebrand disease. These two factors actually bind to each other and circulate in the blood. The factor 9 is Christmas factor. It converts to factor 9A by factor 11A. Its absence causes hemophilia B. Factor 10 is Truart power factor. It has a molecular weight of 59 kilodalton. It converts to factor activated factor 10 by factor 9A, factor 8A, and calcium complex which in the intrinsic pathway, and factor 7A, third A, and calcium in extrinsic pathway. Factor 11, that is plasma thromboplastin. It has a molecular weight of 160 kilodalton. It converts factor 9A. It converts to 9A by factor 12A and activates factor 9. Hegman factor, that is 12th factor, activated by cal kallikrein and kalinogen. In contact with stage surface. It converts factor 11 to factor 11A. The blood coagulation factor 2, 7, 9, and 10 are proteins that is synthesized in the liver. The catabolism of these proteins also happens in the liver. These all uh, blood coagulation factors have a gamma carboxyglutamate residue in their initial portion of the molecules. The gamma glutamate residues are calcium chelators and they helps in the binding of the protein to the phospholipid which is present in the surface of the platelets. The formation of GLA residue by post-translational modification of glutamate by carboxylation requires the presence of vitamin K as a cofactor. So, the lack of vitamin K also can affect the blood coagulation method. With this, we finish about the blood coagulation factors. In the next video, I will explain about how the blood coagulation mechanism